So yesterday we started talking about manipulating DNA, section 1 in chapter 9, and we said that what is the meaning? Who can tell me what is the meaning of manipulating DNA? What is the meaning of manipulating DNA, little guys? In your own words, can anyone tell me what is the meaning of manipulating DNA? No one remembers? Yes, Jane.
It's like changing or controlling the DNA.
Changing or what?
Controlling the DNA.
Yes,
Like,
controlling, changing, playing with it, okay, by changing it. So, why do you think we manipulate the DNA? Why we change in the DNA? What do you think? Why? Why we manipulate in the DNA? Okay, guys, let's just review together. We said that our chapter is talking about the frontiers of biotechnology, and that means what is the last uh, board that the science reached in the biotechnology, in biology, and specifically in the technology in the biology. Okay? The
اوكي.
scientists, yes, yes, Muhammad.
لا لا بس احكي له اوكي.
Ah, so our lesson today will show us how this uh, the frontiers happen through the biotechnology and how the scientists go through more details on the DNA and how they can manipulate it. Why they need to manipulate it to change the DNA of some organisms. يعني في عندي organisms like some plants, animals, and also humans. They have some problem or some traits that are يعني not wanted traits. So in using these techniques اللي هو biotechnology, they can change in the DNA to get better traits, such as this cow, as you see here in the figure. How a cow can produce more milk. How it can produce more milk? By changing its DNA, by manipulating its DNA. Hagel corn is looking very fresh and none of the seeds are affected by any insect. Why? Because they injected with something that was manipulated the DNA, changed the DNA in it, some genes in the DNA, not all the DNA, genes in the DNA. And it becomes more resistant to viruses and bacteria. Also, sometimes manipulating DNA will produce much crop, much food. Do you understand the idea? So, biotechnology, we said, is the use of technology to change the genetic makeup of living things for human purposes. Okay? And generally, the goal of biotechnology is to modify organisms so they are more useful to humans. For example, it may be used to create crops that yield more food or resist pests or viruses. So, how is biotechnology and why they use it? To change the genetic makeup, to change the genes of the living things for human purposes, to modify some genes in animals, in plants, and many organisms for our purposes. And also sometimes this biotechnology can be used to control some disorders in the human. Okay, you understand? Okay, 1 minute, guys. I will mute you all for 1 minute. Okay, guys. So did you get the idea why we what is the biotechnology and why it is used? طيب, if you want to change in the makeup of the living things by changing the genes of the DNA, so we have to manipulate the DNA. How we can manipulate the DNA? By cutting it. Okay? So, and the scientists, we said they use several techniques to manipulate DNA because it is so far too small, it's so far to see, it's so small to see. وصح, large molecule, but it's still too small to see and to handle with your hand. فعشان هيك we use some tools to handle it by chemicals, by computers, and by bacteria. And we said in these tools, they are used in genetic research and biotechnology. Till here, you are all got the idea? Any question till here?
No.
طيب. So why would the scientists want to cut the DNA? Why? لأن ال DNA قلنا هو, to answer the question, you have to remember that a gene is a sequence of a DNA nucleotide. وقلنا كل chromosome is made of a DNA molecule. اوكي؟ كل كروموسوم عبارة عن DNA molecule. طب أنا ال DNA molecule هذا large فيه many genes وهذه الجينز عبارة عن ايش؟ عن DNA nucleotide, sequence of DNA nucleotide. اوكي؟ and it's very long. So a whole chromosome is too large to study a particular gene easily. كتير طويل هو بالنسبة للجينات اللي فيه. It's a big molecule. So they had to find a way to get much smaller pieces of DNA to study genes, pieces of genes alone. Okay, so why do we want to cut the DNA? Our scientists, why they want to cut the DNA, guys? Yalla, why the scientists need? Yes, Murad.
Because the DNA is made of sequence of nucleotides, so they want to cut it so they can change the nucleotides.
لأنه it's a long, very long. Okay, وهذا ال DNA is made of many genes. Yes, يا محمد.
لا miss, I just wanted to answer.
Okay, it's very long and this made of many genes. Genes هي عبارة عن nucleotide sequences. طب أنا if I want to study genes, okay, I cannot study the genes in a whole long DNA. They will cut it to parts, pieces. كل piece it will represent a gene or a specific nucleotide sequences. Okay? So to study a specific gene or a particular gene easily, the whole chromosome is too large. So they had to find a way to get it much smaller pieces of DNA to make it easier to study these genes. هلا قلنا إنه splicing the chromosome into pieces is not like splicing anything else because it's very small, because we cannot see it in our hands. It is a large molecule at a molecular level. يعني لو قارناه the DNA مع other molecules ثانية هو large, but still we cannot see it. Okay? So we can use something is called restriction enzymes اللي هو molecular scissors. يعني هم زي scissors بالضبط بس ولكن they work or we cannot see these scissors because they can cut the DNA at a molecular level يعني على level الجزيئات. We cannot see these. By instead of using a pair of scissors, normal scissors, we cannot use it. Scientists use enzymes that act as molecular scissors, and these enzymes slice apart the DNA, come from many different types of bacteria. منين بيجيبوا هاي الانزيمز restriction enzymes to cut the DNA؟ من البكتيريا. طب من البكتيريا منين بتجيبهم؟ البكتيريا احنا قلنا inside the bacteria إذا صار في عندي أي infection by a virus اللي هو البكتيريوفاج، الفيروس راح يدخل الدي ان اي تاعه inside the bacteria. تمام؟ هلا البكتيريا هاي راح تعمل fighting، بدها تدافع عن حالها، صح؟ it want to fight against this virus DNA. شو بتعمل؟ ربنا خلق لها enzymes اسمهم endonuclease أو restriction enzymes، ممكن تستعمل هذا النيم أو هذا النيم. هدول الانزيمز they have the ability to destroy the DNA of the virus. دمره. اوكي شو بتعمل هاي الانزيمز؟ بتعمل cutting لل DNA pieces. ال DNA is cut to pieces in which it will enable the virus to replicate. DNA تاعت الفيروس replicate. وهيك هي بتكون دافعة عن حالها البكتيريا. Okay? So restriction enzymes act as a molecular scissors, come from various types of bacteria, and these come as a result of defending against the viruses. هلا هدول الانزيمز help the scientists to study and manipulate the genes. استعملوهم الساينتست أخذوهم من البكتيريا extract them from the bacteria. Why? To cut the DNA of other organisms. Okay? لهون تمام واضح من اجت restriction enzymes. Please tell me again. What are the restriction enzymes and what is the use of these restriction enzymes? Yes, Muhammad.
Miss. Restriction enzymes act as the molecular scissors. They come from various types of bacteria. They allow scientists to more easily study and manipulate genes. They cut DNA at specific nucleotide sequence.
هلا حنحكي عن هاي. cut DNA at specific nucleotide sequence. طب from where these enzymes come to the bacteria? يعني ليش البكتيريا بتستعملهم؟ Why the bacteria use these enzymes?
Yes, they are like its immunization system, like the
defending صح.
immune system.
Defending against what? الفيروس DNA. لما الفيروس release the DNA inside the bacteria بتعمل defend تطلع هاي الانزيمز to cut the DNA and enable this DNA to replicate.
It's like the white blood cell. But
مثلا defending صح. هلا هذا restriction enzyme ليش سميناه restriction enzyme؟ Why do you think we named this a restriction enzyme? Anyone knows? Why do you think? I know that you don't know, but why do you think? Why these called the restriction enzymes? Yes.
Yes, because they defend the bacteria.
They defend the bacteria. صح. مظبوط. Yes, continue محمد.
لا بس هيك.
طيب مين بده يزيد عليها لجين؟
I think because they stop any further actions. So called restrict.
Restrict يعني بوقف. Restrict the effect of the virus on the bacteria.
Miss, and it cuts the DNA.
Sorry, Muhammad, your voice is not clear here.
It cuts the DNA.
بالضبط. It cuts the DNA بس سموهم restriction enzymes because it restricts the effect of the virus. بتوقف ال effect of the virus by cutting the bacteria. تمام. هلا هدول restriction enzymes how do they cut the DNA? Different restriction enzymes cut DNA in different ways. هلا restriction enzyme هو enzyme. It must recognize a sequence of DNA. يعني ما بيجي بيقطع هيك عشوائي على الدي ان اي. هي الدي ان اي. الدي ان اي فيه nucleotide sequences. كل enzyme how it cuts the DNA by recognizing a sequence of DNA. بيجي بيلاقي السيكونس مثلا هون GAA guanine adenine adenine. It will start to restrict, to cut here. So each enzyme has a restriction site. كل ما هذا الانزيم يلاقي نفس النيوكليوتايد سيكونس بيعمل cut. في عندي انا انزيم تختلف in different ways they cut حسب the restriction sites that they cut on. Okay? في عندي restriction enzyme 1 as you see here in this figure. it recognizes 3 restriction sites على DNA, فit cut it to how many pieces? The 4. 1, 2, 3, 4. Third restriction enzyme recognizes on 6 restriction sites, which cut the DNA to 7 fragments. So different restriction enzymes cut DNA in different ways. How? Each enzyme has a different restriction site. شو يعني restriction site؟ هو a sequence of a nucleotide that will be recognized by the restriction enzyme. مكان ما حيقطع restriction enzyme. So, يلا, who can tell me again, what is a restriction enzyme? What is a restriction site? Murad.
Restriction enzyme is an enzyme that acts as a molecular scissor to cut the DNA.
Bravo.
A restriction site is a sequence of nucleotides that the restriction enzyme recognizes to cut the DNA.
Bravo. Thank you. إذا كتير guys, any question till here? حدا عنده سؤال لهون؟ Please, محمد, any question?
لا مس.
Okay. هلا أحيانا هدول الكات لما يجي السكشن انزيم بيعمل كات بيعمل ستريت كات هيك مقطوعة ستريت. اوكي هاي بنسميها بلانت اند. تكون الدي ان اي فيها بلانت اند يعني هيك زي قطعة كده بتقطع بالسكينة انت كات ستريت. هاي بنسميها بلانت اند زي هاي. وفي عندي other restriction enzyme بيعملوا staggered cuts. كيف يعني staggered زي هاي؟ بقصوا بالطريقة هاي not straight فبيطلع عندي في tail للنيوكليوتايد exposed tail يعني مش ماسك بأي إشي جاهز إنه يمسك بأي نيوكليوتايد. فعندي restriction enzymes تعمل straight cut وبتعمل blunt end وفي عندي some enzymes تعمل staggered cut and leave sticky end. the staggered cuts زي هدول بتخلي في عندي exposed nucleotide tail of nucleotide مع sticky end. ليش بنسميهم sticky end؟ لأنه it's very easy to stick to another complementary nucleotide. إذا لقوا ال complementary nucleotide اللي يمسكوا معهم على طول راح تمسك بسهولة. So scientists they use more the enzymes that make staggered cuts because staggered cuts will make it easily for genes to hold together by making hydrogen bonds. يعني زي هون عندي أنا GC و CG هدول sticky end. أول ما الجي سي تلاقي CG على طول they will join together by forming hydrogen bonds. وهذا it will make it easier for the pieces of DNA to join together. Okay? So what are the two types of cuts that made by the enzymes? Who can tell me, Yusra? Yusra? Can you hear me, Yusra? Hello? طيب عبد العزيز.
Yes, miss.
يلا يا يسرا. Tell me the two ways that the restriction enzyme cut the DNA.
Some cut straight across the left plant end, and some make a staggered and cut the leaf sticky end.
So which one is more useful do you think? The cut with straight blunt end ولا الcut اللي بيكون فيها staggered end زي هدول بيكون فيها sticky end? Which one is
I think the second one.
second one. برافو. هذا restriction enzyme زي عندي مثلا example of restriction enzyme نسمينه TACI. هذا cut DNA when it finds its restriction site TACI's restriction site اللي هم هدول TCGA AGCT. هذا الانزيم اللي اسمه TACI لازم يلاقي restriction site اللي هو ايش؟ TCGA. فقط بيلاقي هيها TCGA كان هون TCGA لما يلاقيه بيعمل cut عند التي. اوكي؟ لا كمان مرة TCGA بيعمل هذا الانسيكون انزيم كات هون. وهدول الtails will make these pieces with the sticky end, which will make it easier for the pieces of the DNA to stuck with its complementary nucleotides. طيب هلا after a long DNA has been cut by restriction enzymes into many small fragments, several things can be done with the DNA. عملنا cutting the DNA. How we can use it? For what purposes we can use it? شو رأيكم؟ يلا أعطوني examples. Why we can use, how we can use it. ليش عملنا cutting? To study some genes, to use some genes. طيب أعطوني examples for the uses of the DNA after cutting it. عملنا له cutting. يلا. Who can tell me some examples? Yes يا محمد. لجين.
الو مس.
بسرعة مس يا.
Should I say?
Yes, لجين say.
Okay. I think the deletion of a gene, for example.
مثلا برافو إذا كان في عندي بالجين deletion برافو. مراد.
Yes, miss. Can you hear me?
يلا قول لي why we can use it. عملنا cut. شو استفدنا؟
We could use it maybe for some يعني studying for it.
صح studying
and
صح. أنا بدي أدرس جين معين على الدي ان اي، عملت له cutting أخذت الجين اللي بدي إياه.
الو مس. الو
محمد.
اوكي مس سامعيني؟
هلا سمعتك
تمام مس. كمان نقدر برضه replicate live things صح؟
شو إيه؟
مس we can replicate live things.
شو replicate؟ I don't
بس
understand.
يعني replicate للانيمالز يعني.
فهمت عليك. طيب so for example the DNA sequences of genes can be studied. بعدنا عملنا لها cut. اليوزز تاعتها إنه we can use it for several things. أول شيء ال DNA sequence can be studied. ندرسها هذا الجين نشوف وضعه تحت الميكروسكوب. or a gene cut out from DNA can be placed زي ما قصده محمد يحكي can be placed from the DNA into the DNA of another organism. ممكن آخذ جين بصفة من organism من مثلا plant وأحطها في plant ثاني عشان أعمل changing وأعمل better trait. زي مثلا الووتر من البطيخ فيه بذر. ممكن آخذ أنا جين أو DNA sequence from a plant that have no seed وأحطها وين؟ بالدي ان اي تاعت الووترميلون and this will make change بالووترميلون DNA for producing no seed. Okay, yes. Yes, مراد أو محمد, what do you want?
بس لا ما عندي سؤال.
محمد, you have any
أنا ما عندي
question?
سؤال. لا.
Okay, so يا إما for studying the gene أو for وزي ما قالت لجين study the genes بقدر أعرف إذا في mutation أو لا في هذا الجين. Okay? And الجين cut out from the DNA ممكن أخذه أنا out from the DNA from one organism and place it into the DNA of another organism. But before anything else can be done, قبل ما نعمل هذا كله, the DNA fragments have to be separated from one another. أنا عملت لهم cutting. أنا هم عبارة عن mixture. الدي ان اي صح كامل بس مقسم لبيسز مخلوطين مع بعضهم الفراجمنت. I have to separate these DNA fragments from one another. كيف نعمل separation للفراجمنت؟ This is what we are going to talk about now by using a technique اسمه gel electrophoresis. Gel electrophoresis هو عبارة عن gel used to separate DNA fragments by size. الآن عندي الفراجمنت تقسمت. الدي ان اي تقسمت. حطوا restriction enzyme. كل restriction enzyme have a restriction site وبيطلع عندي البيسز with different length. اللينك بتاعتها مختلفة عن ال DNA pieces. فهذا الالكتروفوريسيس عبارة عن تكنيك يعمل separation لل DNA fragments by size. يعني بفصلهم عن بعض عن طريق السايز الأصغر الأكبر للأصغر للأصغر. أوكي؟ هلا شو بكون عندي هون؟ I want to show you the electrophoresis first. هذا we have to watch this video now to see what is electrophoresis and how does it work. Let's watch this together. عشان نفهم شو هو electrophoresis. Can you see the video?
Yes.
يلا we will start.
This kind of an image is not new to us, especially for people who love forensic science. But what exactly is this? It's an image of the separated DNA fragments on a piece of gel. And the process of separating them is called gel electrophoresis.
يعني هذا how the gel electrophoresis هلا we will see the steps how it works and how it's made. هدول at the end احنا بنشوف ال DNA fragments as bands على قطعة جل. كيف الجل بيعملوا على قطعة جل بيخلوا هدول ال DNA fragments to move and تعمل band وبيطلع عندي هذا الماب that we will study العلماء بدرسوه بيكون عبارة عن جل عليه bands of DNA. تمام؟ هذا معناته إن DNA fragments were separated. Let's see how.
Doesn't this sound very difficult? Separating DNA fragments on a piece of gel? But actually, the process of gel electrophoresis is not very difficult. In fact, it's quite easy. Let's have a look at how the process works and also the principle behind it. Let's begin with the definition first. Gel electrophoresis is a technique to separate the different DNA molecules based on their sizes.
شايفين؟
Can you get
دايما تحطوا في بالكم هو تكنيك بيعمل different DNA separate different DNA fragments على أي أساس؟ على أساس السايز. أوكي؟ بيعملهم separation على أساس السايز.
guess why is it called gel electrophoresis? Okay, let me help you with this. This separation technique is based on the movement of charged molecules when exposed to an electrical field. And this movement occurs in a gel medium. Hence, it's known as gel electrophoresis.
إذا ليش سموه electrophoresis gel electrophoresis؟ لأنه عندي التشارجد موليكيول اللي هم الدي ان اي. هلا الدي ان اي مين بيعرف هو بوزيتيف ولا نيجاتيف موليكيول؟ ايش التشارج تاعته الدي ان اي؟ بوزيتيف ولا نيجاتيف؟ Can you guess؟ Yes, محمد.
Miss, repeat your question.
What do you think the charge of the DNA? هو positive ولا negatively charged؟ إياد.
Miss, positive.
إياد شو رأيك؟
I think positive.
لا هو negative. عشان الفوسفات جروب اللي فيها negative charge وهي هاي اللي هي exposed تكون برا طالعة على الباك بون فبتعطيه negative charge. اوكي؟ تمام؟ فهو الدي ان اي عبارة عن negative molecule. هلا هدول الموليكيولز اوف دي ان اي اللي هم الفراجمنتس اوف دي ان اي صاروا pieces they will move. اوكي؟ حيمشوا وين؟ بجل. هدول الفراجمنت حيمشوا بجل موف جل وهم تشارج موليكيولز فيهم تشارج نيجاتيفلي تشارج. كيف بدهم يمشوا؟ عن طريق electrical field بيعملوا هون تيار كهربائي موصول بهذا الجل to let these charge move. Look how.
Now let's understand the process step by step. But before that, since the term charge is used by us here, can you guess the charge present on a DNA molecule? Let me tell you that a DNA is negatively charged. And this is due to the presence of phosphate groups. These phosphate being negatively charged and present in the backbone of the DNA double helix impart a negative charge to the complete molecule. And what is the gel used? Interestingly, the gel which is generally used for the DNA is agarose gel, and it's obtained from the seaweeds. Now, getting back to our process. We said that the DNA molecules get separated on the basis of their sizes. So, if we have a sample of DNA which is containing fragments formed with the activity of restriction
أنا عندي هاي السامبل هون، أما جبت أخذت الإنزيم على السامبل تاعت الدي إن إيه وعملت لها كاتنج، فالسامبل تاعت الدي إن إيه فراجمنتس بتكون كلها مخلوطة مع بعض. وين بهذا الأمبول، أوكي؟ هو الأمبول، هاي السامبل أوف دي إن إيه.
enzymes, then separating the fragments based on the difference in their sizes would be possible with gel electrophoresis. Let's have a look at this in detail. To understand the principle and working, let's take the analogy of a sieve. We know that if a mixture of different sized particles is poured onto a sieve, then only the particles smaller in size than the pores will escape, while those bigger in size will be retained back. Here, the size of the particles is the only parameter used for the separation. Same is the case with DNA fragments. When our DNA sample containing various size fragments is loaded onto a gel, then the charge applied across the gel helps in the mobility of these fragments. However, not all fragments move at the same pace. Those which are smaller move ahead, and those which are larger in size find it.
إذا حطينا السامبل بتستعملوا على فكرة هم بالجل، بتجيبوا الجل بتستعملوا كوم، كيف المشط مشطوا فيه كوم، يعملوا خلوق بالجل، اوكي، وبحطوا السامبل اوف ذا دي ان اي هون، مثلا ما بكون عندي 1 سامبل هون باخذوا دي ان اي سامبل من اكثر من سامبل بيشغلوه مع بعضه في الالكتروفوريسيس. هذا بحطوا السامبلز هون، كل دي ان اي رح تتقسم لباند. هلا اللي بيكون شو رايكم مين بيكون اسرع؟ الباند او سوري البارتيكل الفراجمنت اللي بتكون سمول لينث ولا لارج لينث؟ مين حتكون اسرع؟ الديستانس حتمشي اسرع. شو رايكم؟ يس محمد.
بس مين راح تكون اسرع، اللارج
ولا السمول فراجمنت؟
مس السمول.
صح، كل ما كانت اخف كل ما كانت اسرع. فهلا هذا الجل الكتروفوريسيس هو عباره عن جل حطوا فيه فتحات عن طريق كوم مشط عادي بيفتحوا فيه فتحات، بحطوا الدي ان اي سامبلز هون، وبيكون الدي ان اي اصلا هي نيجاتيف تشارج، وهذا الالكتروفوريسيس الجل بوصلوه مع تيار كهربائي فيه نيجاتيف الكترود وبوزيتيف الكترود. تمام؟ هلا بما انه الدي ان اي هو عباره عن نيجاتيف تشارج، لما نوصل احنا الكرنت الكهرباء، اوكي، الالكتريك كرنت، رح تمشي الدي ان اي من نيجاتيف لبوزيتيف لان هي نيجاتيف، حيصير في تجاذب بين النيجاتيف والبوزيتيف. سو لما نبلش احنا نشغل الكهرباء نوصله بالكهرباء، هذا حيخلي الدي ان اي fragments to move from negative pole to positive pole. طبعا الدي ان اي عباره عن fragment اللي حتكون الفراجمنت smaller in size او باللينث بتكون اقصر، it will move faster than the bigger fragment لانها اخف.
Difficult to move. To understand this better, let's zoom into the gel that is prepared for the experiment. Here, at the microscopic level, we find that the gel appears to be a mesh-like structure and the pore size is nearly constant throughout. So now imagine what will happen if different sized DNA fragments are made to pass.
نفس الشيء كيف انتم شفتوا المصفاية كيف عندكم انتم البيج particles والثيك end والسمول particles can move، نفس الشيء عندي بالجل هذا، بس you cannot see it in your eyes but under a microscope it's like a mesh، زي كيف المصفاية عاملينه فيه زي pores، يعني زي فتحات صغيرة but you cannot see it in your eyes. هدول الفتحات بيكون عندي انا البارت او الفراجمنتس اوف ذا دي ان اي اللي هم with bigger size cannot go through it. It will stuck inside. رح توقف فيها، رح تمسكها. اوكي؟ زي المصفاية بالضبط. تمام؟ واضح مش فاهمين؟ اما السمولر sizes will move through it، خلص رح تمرق منها. We understand guys till here? في حدا عنده سؤال لهلا؟ يس محمد.
لا, nothing.
Okay.
Pass through these pores? It's obvious that the small size fragments will escape the pores faster, while the larger fragments will find it difficult to come out. They will take a lot of time to cross the smaller pores and thus will lag behind. This is how we can understand the principle of separation of the DNA fragments based on their sizes. Now let's see how the procedure works. The first requirement is the complete setup. This will include the casting tray, gel, a comb for making wells in the gel, electric supply, and most importantly, DNA sample with different sized DNA fragments. Here's an illustration of the casting tray containing the gel in which we will load the DNA mixture. We usually opt for a comb to form well-like structures into the gel. And why do we do that? Because it's in these wells in the agarose gel that we load the DNA mixture.
شايفين هي الجل فيه نيجاتيف وبوزيتيف الكترون تعمل كوم عشان يعملوا ويل فتحات في الجل عشان في كل فتحة تحطوا different DNA sample.
Tell me, where should the wells be formed? At the ends or at the center? Think about it. We know that the DNA fragments, being negatively charged, will travel from a point of negative charge to the point of positive charge. Thus, we need to form the wells to load the DNA samples near the negative terminal or the cathode to be precise, so that the DNA will move towards the positively charged anode. This movement of the DNA molecule will be promoted by the electric field. But we cannot track the movement of the DNA in the gel as it's colorless. For this, we use a colored loading dye to track the movement of the DNA.
نستعمل dye عشان track the movement of the DNA.
The mixture of the DNA samples with the colored loading dye is now all set to get into the gel. But before loading the DNA mixture, we will fill the apparatus with a buffer as shown. The buffer is used to provide better conductivity of electricity. Next, we will load the DNA mixture into the wells and we're ready to turn on the power supply. Once the power supply is turned on, the DNA mixture will move through the gel, which can be tracked with the help of the loading dye. The loading dye is selected in such a way that it travels a bit faster than the DNA segments present in the mixture. And why is that so? That's because...
Okay guys, we will stop till here now. Any question before I leave? Tomorrow we'll continue. Any question guys before I leave? Yes, Muhammad?
مش ما عندي.
Any question?